Alright, in this video we're going to go over cell biology. Alright, and we're going to start with cells, kind of like overview of what the individual cells do and basically what their parts do. The major parts are going to be called organelles. So just as you have organs in your body, you have that, and these organs have certain functions. Cells have certain organelles. They're little structures inside your cells that also have some type of function. And theirs is much more chemical in nature. They're very tiny, of course, but the analogy with your organs is very similar. You have a stomach, for instance, which is designed to digest something, and you have things inside your cells that are also designed to digest something like a lysosome or something. All right. So here we're also going to talk about some cancer. So I'm going to go with the cancer here and start talking about that very just briefly here. Let me enlarge this a little. Let me talk about cancer. And what is cancer? Well, cancer is simply a confused cell. We've already learned that, for instance, the DNA, your genes, all right, they are the instructions for making proteins. Okay, we should have learned that already in previous videos. So you have genes that are instructions for making proteins. What do the proteins do? The proteins, they do all the chemical work in your cells. So the idea here is also that the cells, each cell does not use all the genes. It only uses some of the genes. So let's write that down as well. Oops. Here we're going to say that in a multicellular, we are multicellular. We have many cells. Multicellular organism all right, in a multicellular organism, let's get this spelling right. Not all genes are used in every cell. In fact, that's what makes one cell type different from another. Different genes turned on, other genes turned off, that determines your cell type. So the genes that are on, all right, determine the cell type. And actually, no cell should have all genes turned on. That would probably kill it. But you can have cells that get the wrong combinations turned on, or turned on at the wrong time. So cells that have the wrong combination of genes turned on, essentially this is cancer. All right, let me put an equal sign there. I'll put two equal signs. So in other words, if you have the wrong combination of genes turned on, you're going to make the wrong what? You're going to make the wrong proteins. And the proteins control the chemistry of the cell. So, and the cell type. And so what happens is if you somehow get the wrong genes or the wrong combination of genes turned on in a cell, say there's a protein that does something that makes you a skin cell, but you get that turned on inside of a liver cell, well, that liver cell isn't going to work right. And essentially, this is what we call cancer. Now, it might not be malignant cancer. Malignant means it can move around different parts of your body and grow and reproduce faster than it should. There are lots of things that make it malignant, which means you're going to have a tumor or something that you're going to have to worry about. You have to go get radiation or therapy or something, go on chemotherapy. Those things, all right, those types of cancers are different than your typical cancer or your typical confused cell. We all have confused cells. Things like warts, all kinds of things are confused cells. They're not dangerous, all right? They could become dangerous if they start growing out of control, and that's when we would call it cancer. But ultimately, this happens a lot. And actually, what you also need to know that I haven't written here is that you have mechanisms in your cells, in your genes, that check on to see if your cells are doing the right thing. And if your cells are not doing the right thing, there are mechanisms for killing that cell. In other words, the cell will commit suicide if it becomes cancerous and is aware of it. There are chemical reactions that should not occur together, and if they do, it stimulates some other genes to be turned on, and the cell essentially lyses. It literally blows up, if you want to think of it that way. So that's why you don't see cancer until you're much older. Over time, what happens is things that cause mutations, that would be changing your DNA, all right? Changing your DNA alters the proteins that are made in your cell, so there maybe you're making the wrong protein, but it can also alter which cells are turned on and off. And if you turn off the cancer-preventing cells, those are the ones that tend to, I should say, the suicide cells that allow the cell to kill itself when it's aware that it is not doing the right thing. When you destroy the suicide genes, that's when you really are in a high risk of getting cancer, and that occurs usually later in life, after you've had a chance to accumulate lots of mutations. And the problem is a cell that loses its ability to kill itself is the worst type of cancer, and at least your genes will help you fight the cancer if those are still present. But as a piece of trivia, cigarette smoke. Actually, any kind of smoke from burning foliage, like burning leaves even, but tobacco is really bad, produces a chemical, and that chemical actually targets the suicide genes and damages them. And so that's why cigarette smoke in particular is harmful to you and increases your risk of cancer. If I have time, I'll go into detail on that. I won't have time to do that in this video. But I did want to throw that in just so that you have that. And might be a short question on what is cancer. And you should certainly include the topics that I've written down here. But you also need to phrase it in a paragraph, and so you should have listened to this and taken notes on what I just said. So not just these facts, but put it all together into a long paragraph, and that will, I would say, 100% chance that that's probably is going to be on the test. So I'm going to ask you what cancer is in a very general way, and I want you to be able to tell me what it is. It's a confused cell, but you need to add some of these things in there with it. All right, moving on to the regular part. What do you need to know? Well, I'm not concerned about you knowing these people. You do need to know that all organisms are composed of one or more cells. Generally, we are eukaryotes, and they are typically multicellular. So we are multicellular organisms. We've got lots of cells. Let's see, maybe 3 billion in a 100-pound person, something like that. Don't quote me on that, but that sounds about right. That's if I remember correctly. So we've got a lot of cells, and we have several cell types. And of course, as I mentioned, what determines the cell type, what makes an eye cell, or skin cell different from a liver cell or from a neuron, they have different genes turned on and off, which means they make different proteins, and the proteins do the chemical work. So that means they are chemically different, and that's what gives them their shape, their structure, and their abilities. All right, so cells are the smallest unit of life. You can only have single-celled organisms. Nothing else is living that's smaller. Viruses are not alive. Um, they might come close sometimes, but they are not living. So you don't consider viruses alive. But a bacteria is. A bacteria is a much larger thing, and it's much more complex. We usually call those prokaryotes. You might have heard of that term. We are eukaryotes, which are the multicellular, or I should say, typically multicellular. There are no multicellular prokaryotes and no multicellular bacteria, though they do live in colonies sometimes. All right, and the last thing you need to know here is that new cells come from pre-existing cells by cell division. In other words, life is thought to have started with some original organism and multiplied and essentially became all the life that we see on the planet. Let's see. Moving on. We already know about nucleotides, DNA, and amino acids. You pretty much go DNA, RNA, proteins. You should be familiar with that with some videos that I have that I've made. These three molecules. We've talked a little bit in some videos about membranes. We will talk more. And we also need to know that these polymers are enclosed in membranes that give something cellular properties. So what are cellular properties? Well, we go on. Essentially something that's compartmentalized. So that's a good word to know. Let's switch back to our thing. Let's scroll down here. You need to know this term, compartmentalization. Compartmentalization here. And it's what you might think. Think in terms of a house. A house is compartmentalized into rooms. Has rooms that have special function, that have a specific function. Oop. All right. Should make that plural, I guess. Well, just like that, so do cells. They have compartments that we call organelles. Organelles are compartments, like rooms, that have specialized function. So keep that in mind. You can think of it as a house with rooms. You have a house where you do your cooking. That's called the kitchen. Well, you have something kind of like that in the Golgi apparatus or in the endoplasmic reticulum. Not sure which one I would count that as, but you do certain things, you package things, do things like that in certain organelles, just like you might do that in the kitchen where you might be doing some cooking or something. You have a place where you sleep or watch TV. Well, you have mitochondria or chloroplasts that do specialized functions within the cells. Now, one key idea here is that there are no, generally, we're going to say there are no organelles in prokaryotes, like bacteria, single-celled organisms. One of the key factors is eukaryotes. Multicellular organisms are the ones that typically have the organelles. All right. Now this makes the prokaryote, the bacteria, very simple, and it can't do as much. Without compartmentalization, it's limited on the chemistry that it can do. What do the compartments let the eukaryote do, these little organelles? Well, they can do some fancy chemistry that won't interfere with the regular chemistry in the cell. So, if you want to do something like watch TV, but if somebody is like frying bacon, you can't hear the TV. They interrupt each other. So if you were living like in a little apartment and they're like a motel, someone's over cooking something, making a lot of noise, you can't hear the TV, they interfere. So that limits what prokaryotes can do. They can't do the chemistry, certain types of chemistry at the same time that would make it more efficient. Whereas eukaryotes have organelles, and they can compartmentalize this chemistry into these little compartments and keep the chemistry that's going on inside one part of the cell separate from what's going on in another part of the cell, allowing them to do a lot fancier chemistry is what I would, it's one way to say it. All right, so keep that in mind. That's something else that you should know. And the walls, you might say, of these little compartments are membranes. All right, membranes allow us to compartmentalize. And this polymers, the DNA, proteins, RNA that are enclosed in these cellular compartments that I'm talking about are in membrane, separated by membranes. That gives the individual cell its properties, its structure, and its function. All right, so here we need to learn a little bit about, I'm not going to ask a lot of questions on this, but you need to know that there are different theories on how life began. I'm going to let you read these different theories on your own. There will be multiple choice, true, false. I'm probably not going to ask you any essay questions on this, but you might get some true, false, and some multiple choice on these various theories of the origin of life. Maybe know about some of the experiments, know, maybe why you need some heat or some certain chemicals, like in a volcano, in a vent at the bottom of the ocean, or know what organic polymers are. The organic polymers we are interested in, polymers are long chains, and we've already heard of DNA and RNA and proteins, and those come from certain types of environments where those can form naturally without any outside influence like a human in a chemistry lab. They would form naturally, and if they come together in the right combinations, it's thought that they potentially could have created the first life. No one knows that for sure, though. So you need to know these, know about what they're talking about, polymers, and prebiotic means pre-living, all right? But these things that we can create, these things in a test tube, but they could have occurred in nature also. Now the thing is, we can create these chemicals, but we've never mixed them up in such a way to form life, although people are trying. All right, boundaries would have had to have been created, very similar to the organelles creating the compartments. So something like that would have had to have occurred as well. Probiotic liposomes. I probably won't ask you anything, but you need to read about it. I might ask you where you kind of have to know knowledge of this and realize that these things form naturally. This is not something that life created. These little membrane-bound not organelles, but spheres. They're phospholipid bilayer is the name of the kind of the material here. We've already talked about membranes, but that's just a fancy word for membrane, phospholipid bilayer. And they usually are filled with water. Be a perfect place to start a cell. All right, now it's thought that RNA was probably the first molecule to be formed, so you needed some RNA to get into one of these little vesicles, along with maybe some proteins and some other things to cause things to occur over and over again. Something that could let certain things got into the little little compartment here, and there was some RNA in there, and these things that got in would do some chemistry in here that might have at some point led to the multiplication of that RNA, and that would have been probably the very beginnings of life. So you need to read about this. I am not going to go over this specifically. So I want you to be aware of it. There might be some multiple choice. This could be a short answer in some of these, so do not skip them. I'm just not going to go over them in detail. Do know that RNA is thought to be the first of the. We can't prove this until we actually create artificial life in the test tube. That would be a big deal. I might give you a little story on, in a way, we already can do this, but not really. It's not enough to say that we can create life, but we can certainly take parts from living organisms and rearrange them very easily. So if you want to consider that creating life, I'll tell you that story later. Kind of scary, but we understand that there might be an argument for other types of molecules to have started life. But as a general rule, because it's a DNA, RNA, protein world, and to understand what I mean by that, you need to have watched the other videos. You'll realize that this is transcription and translation, and they are what this exam is about. So you do need to know a little bit about this, because it leads to this idea of where life comes from by understanding transcription and translation. Now here's some things you need to know just in general, maybe true-false questions. But of course, where do you store the information on how to make your cells? Well, it's in the DNA. So you store that information in DNA. Now, what kind of information is stored in there? Well, it's how to make your proteins. But the DNA are not the actual instructions. Each little gene is copied from the DNA, and because the DNA is protected, it's protected in the nucleus, and you have to make a copy of it, and that copy is what is used to a copy of the instruction that's what the ribosome reads in order to form the protein. So the information is stored long-term here, but you make a short-term copy, send it to a ribosome, and the ribosome is a little machine. It's a chemical machine, but it has the ability to read the RNA and follow the instructions. All right, and because the DNA is protected, it is less likely to get damaged and have mutations, and that's important. You don't want it to be damaged, because we already talked about mutations causing cancer, the cell doesn't work very well, that sort of thing. Now you also kind of need to know what metabolism is. Metabolism is just the chemistry needed to carry out the cellular functions, like how to make membrane, how to digest food, how to move it around and put it in the right spots, that sort of thing. Now microscopy, I am going to leave you to read this. I will ask some short answers, some true falses, and some multiple choice on microscopy. If you've used a microscope before, you probably know some of this, but I do want you to know the difference between a light microscope and an electron microscope. In fact, that sounds like a very good essay question. What is the difference between a light microscope and an electron microscope? In other words, you might not just tell me the resolution and everything, but how do they work? What is the difference between using just regular light and then using a beam of electrons? Why is that important? What does this let you do that you can't do here? So I will let you read these, and so I'm going to leave that up to you to read, all right, on the microscopy. All right. Now we come down here, and this is where the main part of this one is, and that's on cell structure and function. Matter, energy, organization, probably won't ask you that, but you probably should know that proteins interact with each other. There are, you make these little chemicals called proteins, and they're all different shapes and sizes, but they interact with each other chemically, and they also work to create the cell structures inside your cells, the things like the organelles or the ribosomes, or even making the cell membrane. All of that is done, all of that kind of work, chemical work and physical work, as in they move. These proteins are not static. You add what's called ATP to them, and ATP is to the cell what gasoline is to your car. All right, if you add ATP to the cell, it means the proteins can change shape and usually do what they need to do in order to keep you alive. You run out of ATP, your cell dies. Now, where do we get ATP? Basically, it's what we turn carbohydrates into. ATP can be created from glucose. And so you eat complex sugars or complex carbohydrates, and you can actually convert those into glucose, and then glucose can be turned into ATP to power your cells. So you need to know that series of things I just said there. I've said them before, but you need to know those. Now, I've already mentioned prokaryotes and eukaryotes. These are the more complex ones. They have the compartments, the organelles. This is very simple. Prokaryotes are the bacteria. They're very, very simple. Know that eukaryotes have organelles, and they have a nucleus where you store the DNA to protect it. So read about these. You need to know that, of course, bacteria are smaller, and just know these kind of facts. Am I going to ask you the exact size? Doubtful. All right. You do need to know about archaea. Archaea are extremophiles. They said right here, extreme environments. Extremophiles means they like to live in really harsh, usually what we would call harsh environments, like very high in the atmosphere where there's barely oxygen, or at the bottom of the ocean where there are lots of pressure and no light. And so they usually live in places where nothing else is living except for them. And there are no diseases, I shouldn't say diseases, there are no, archaea have never been known to cause a disease in humans. They are not a pathogen, mainly because they can't survive except for in their very unique area, like in an underground lake under Antarctica. So there's ice on top, but because it's below the surface, it's under pressure, and the water does not freeze, at least not at the same temperature, or it has salt in it, so it doesn't freeze. And they can live in there, and they'll be the only thing living there. And generally, we distinguish bacteria and archaea by their DNA. They look alike. An archaea will look just like a bacteria, but its genes are much different. And because of this, we've decided that they need to be separated from the bacteria as a group. They are, like I said, they are the extremophiles, and they're not very common. You have to go to very extreme places, boiling sulfuric acid at lake, not lake, where the geysers are. I forgot the park in the United States. But anyways, where archaea, there are archaea, living organisms living in boiling sulfuric acid. People fall into that thing when they go. The tourists go look at the geysers, and I mean it instantly kills them. It literally, not only is it boiling, but it's also sulfuric acid. Literally, the flesh dissolves off their bones if a person or an animal falls into the water. It's not really water; it's sulfuric acid. But there are organisms that can live in there, and they are, of course, extremophiles, and they are a type of archaea. One other thing, realize most bacteria don't hurt humans, and in fact you have bacteria that live in your gut that are absolutely necessary. If they weren't there, you would die. So they are called symbionts. All right, and so realize that you've got all kinds of things living on you and in you that if they weren't there, you would die, or you would at least be very sick. We may talk about that. I don't know if we'll have time. But, of course, some of them also make you sick if they get inside of you, like the bad type of E. coli. There's also good E. coli that are necessary for you to survive, and you wouldn't be able to digest your food completely if you didn't have the E. coli. But those are the good ones. You want the good ones, you don't want the bad ones. There aren't many parts to a bacteria. You should read about these in the book. I'm probably not going to say a lot about them in the videos, but these are the things that might be in a prokaryote or a bacteria or archaea. But you need to know in general what these things look like. If you saw a drawing, you would be able to label it, that sort of thing. Might give you a lab on that if we have time. And actually, here is the picture I would probably use. So this would be a very important picture probably for your exam. Now, our cells are more complex. We have compartments, which we call organelles. I might give you a special video on that. And we have lots of different types of compartments. Now, here's an animal cell. It's going to have a slightly different set of compartments than a plant cell. And I do want to say one thing, that you have many types of cells, and all of them are going to have pretty much these parts, but they might be shaped different. They might have more of certain things than others. So they may have a lot more mitochondria in, say, something like a muscle cell, but there might be cells that don't have many mitochondria at all, like a skin cell. So there are different cell types, but as a general rule, these are the parts that you're going to find in an animal cell. Like I said, this is what I'm saying. There are different shapes. So here it's a neuron in your brain. Here's a skin cell. This is much more complex than this. This is going to have not quite so many genes turned on. This is a lot of genes turned on that are very unique to just the neuron. Neuron is a very strange cell compared to all the other cells in your body. Here's your typical plant cell. Now, what you do need to know is the differences between this cell and an animal cell. There are certain things that are only going to be in plant cells, like chloroplasts and a cell wall. All right, so keep that in mind. There are organelles that are not membrane bound. Droplet organelles are separated by things like pH and salinity, and they call liquid-liquid phase separation. And this is a new idea. When I was in school, they said organelles had to be membrane bound, and your typical definition is that it's membrane bound. And I prefer that definition. I would rather them call this something else. But you might consider this, since the book is going through this detail, this liquid phase separation might also cause certain things to not pass or to pass in your one of these structures, and you can call it an organelle at that point. The proteome. Now, the proteome are all the proteins that exist in your cells. All right, and there are different proteomes. Different cells are going to have different set of proteins because they have a different set of genes turned on or off. All right, the proteome largely determines the characteristics of a cell. And of course, proteins, which I've mentioned before, they're going to determine the function and structure. And now remember, one of the themes in this course is structure often determines the function. So if the protein determines a structure, it's probably going to have an effect on the function. And of course, I've already mentioned cancer cells here. The cell surface, all right. Larger cells have a surface area, larger ratio. Know this. You're not going to have to do any math, but know this fact, all right, that the relationship between surface area and volume, all right. Know what happens and what the implications are. If you increase one, what's going to happen to the other one? But you don't have to know the formula or anything or do any math. Just know the relationship, the ratio, you might say, between the two, or how it changes. The cytosol is just the kind of the stuff in between everything else. You have something very like the consistency of jello inside the cells, in between the compartments, like the nucleus and the mitochondria, and there's a lot of stuff going on in there. And we often call that the cytosol, all right? It's inside the plasma membrane, the outer membrane. And you have, sitting in that are the organelles. You have something called the endomembrane system, and essentially the cytosol is all the goop, all right, that's in between them. Would be the yellow or beige looking stuff here. All right, so molecular synthesis. There are certain proteins that are called enzymes, and they do what's called, they catalyze certain chemical reactions. And what that means is they make them happen now. In other words, there might be a chemical reaction between two chemicals that might occur, given time. So you mix two of them mirror each other, and they might get around to reacting, once every 100 years. They're not going to act fast. But if you want it to happen right now at this specific time, then you create an enzyme, a protein that will come along and cause these two chemicals to react in a way that you want it to do right now. Now, it might occur naturally given enough time, but if you want it to happen right now, you make a protein called an enzyme, and that reaction will occur immediately. Now, that can be broken down into two types. So enzymes perform either catabolism or anabolism. There are actually more than just enzymes do this, but essentially groups of chemicals in your body will do catabolism or anabolism. And catabolism is just simply the breakdown into smaller pieces or smaller components. Anabolism is taking those smaller components and putting them back together. So generally we eat something, we do catabolism on it, and then when we build something up and we want to turn basically what we created from catabolism, we want to put it back together into our body. In other words, make new cells and stuff. We call that anabolism. So you need to understand the difference between those. There will be questions on what is an enzyme, what is catabolism, and what is anabolism, and what is metabolism in general. The cytoskeleton kind of holds, it's like the frame of the cell, so you might read about these. I won't ask a lot of questions on these, but you need to be aware of them. So I'm going to leave these for a while. I want you to read over those and know these, but there are certain things I'm just going to leave for you to read out of the book yourself. So motor proteins, really cool. And again, here we have our ATP. I'm going to do a whole video on ATP, I think. And essentially, it allows your proteins to change shape. And of course, if they can change shape, they can do mechanical work, like they can move. I'm going to show a video that's going to show you how they move things around inside your cell. It's really cool. So keep that in mind, that ATP is necessary to allow your proteins to change shape. And there are a group of those called motor proteins that move things around inside your cells. And here it's going to talk about them. It allows your muscles to move, for one thing. Your cells change shape, which then in turn and release certain chemicals, which in turn moves your entire muscle, which is of course made out of cells. These are cool. I'm going to show you a video of these things. So some things just work spontaneously. I'll show you some things that actually don't need ATP sometimes. They just work by other means. Basically, you change the temperature and certain things are going to happen. You change the salinity, that's how much salt is available, or the pH, and certain things automatically happen. You don't actually have to use the ATP to make things work. Sometimes just changes in the environment can cause the proteins to change shape and do some work. You need to know what a flagella is and cilia. You need to know the difference between the two. So flagella, cilia. All right. And let's look here. This is more details on this. I'm not going to ask you details on this, so you can skip that one. I'll just tell you. You need to know a little bit about the network of membranes, the Golgi, the endoplasmic reticulum. If I have time, I'll go back over some of that. You do need to know all about the nucleus, the nuclear membrane. Know that it has pores. All right. That's going to be important later on, so kind of just be aware of it. You read over this, maybe know some general parts, but pay attention to where the DNA is at, where it's not at, and these pores. All right. So I'm going to leave this. know that ribosomes are assembled in the nucleus. The ER. Read this. I will ask some questions on the ER. So I'm going to leave this for you. I might give you a video, but in case I don't, make sure you've read this and understand what the ER does. Also, the Golgi. I'll leave that as open that you should read it and try to understand as well as you can. Know that there is rough ER and smooth ER. Ribosomes are on the rough ER. The smooth ER do not have ribosomes. You don't need to know this about the endomembrane system, and I won't ask you this piece either. So all of this here, you do need to know what a lysosome is, and I will tell you right now. Lysosome is like your stomach, the stomach for your or your digestive system, if you want, in a way. Like your stomach and your intestines are organs in your body. The lysosome is an organelle in the cell that kind of helps you get rid of, digest things and get rid of some wastes. And we'll talk about it. Vacuoles are for storage. This is the one thing that bacteria don't normally consider to have organelles, but there might be a few that have vacuoles that store water, but that would be about it. That's the closest thing to an organelle that a bacteria has, and it's much different there. It's much simpler, and so they really still kind of hold on to the idea that nope, there are no bacteria with organelles. All right, now this is a single-celled organism. All right, so the paramecium is kind of interesting. It has some parts here, but it's still single-celled, and it does have some vacuoles. So keep that in mind. Find out. I tell you what. Find out, is this classified as a eukaryote or a prokaryote? So I would look that up. It's paramecium right here. What is it? Eukaryote or prokaryote? Peroxisomes. They detoxify things. All right. This is an interesting one to read about. Might not get to it on this exam, but it might be worth looking at now just in case. Now the plasma membrane is the one on the outside. It's the big main membrane on the outside of the cell. All right. Know the functions that it has. We'll be going into these in more detail later. All right, know the structure. You must know the structure of the membrane. Know the lipid bilayer, and know that there are proteins stuck in it. So there are proteins with different functions. Some are for cell adhesion, membrane transport, and signaling. So kind of review these three. These are the proteins that are stuck in the membrane, the outer membrane. All right, these are the things that it can do. All right, so there are membranes in the protein that have specific functions. It's not all just lipid bilayer membrane. There are proteins in it. Know that the mitochondria and the chloroplast are unique, and that they have their own DNA. There will be a video that I'm going to post on the endosymbiotic hypothesis, and this is related to that idea here. Where do the mitochondria and the chloroplast come from? Definitely an essay question on the exam. All right, another picture of an animal cell. Know that mitochondria and chloroplasts, their jobs are to make ATP. So they're very good at it. They're specialized. Know that. All right. This is the structure of a mitochondria. You need to know this. You need to know that right there. All right. Know all the parts of the mitochondria. The chloroplast, same idea, but it uses light to make it more efficient. Chloroplast does essentially the same thing that a mitochondria does. It's just a lot better at it because it uses light energy to make it more efficient. You also need to know the structures in this, just like you needed to know the structures in the mitochondria. And you need to know that they have their own genomes. They have their own DNA. So are they living? That's the question. And that might be an extra credit question. Are mitochondria and chloroplasts living organisms? Well, they're inside your cells. What did we say at the beginning of the course? At the beginning of this video, I mean. All right, and I will make a whole video on the endosymbiosis, which will answer that question for you. So you need to make sure you know this theory, and I will have an entire video going over it. Probably won't get to anything on protein sorting. It's cool, but I'm probably just not gonna. We're not gonna have time to cover this. So this will be something that I regret having to skip, but it's not one of the required things we cover. I think it's cool. You should probably know it, but the test can only be so long. Systems biology. No, we won't cover that either. You do kind of need to know that the bacteria is less complex than the eukaryotes. So these are the eukaryotes, and the bacteria are the simple ones, and that is kind of covered here. That is probably a good table to learn. It would help you with some other questions, but as far as me asking specifically about systems biology, probably not. All right, not a bad picture either. You can look over that. That is kind of a test question, I guess. Be a cell, and you got to tell me about the different parts. Where are they? So this is a good review right here. I kind of like it. But that's it for this video. A little bit of cancer, a lot of cell biology, kind of giving you some hints on what you should study. All right.